Thanks everyone for coming this morning to an 8 a.m. talk on harmful algal blooms and hypoxia. So as Greg mentioned, I've been studying these types of events for my entire professional career, 13 years, so I'm not, some people may laugh, and that's okay. But I've studied these issues throughout the Northeast U.S., down along the East Coast, as well as several continents. So we have ongoing work in Africa, New Zealand, China. We have some work going on in Europe as well. So I've studied these types of events on many different continents, and there's a lot of similarities, but also a lot of differences. So today, what I wanted to talk mainly about are the harmful algal blooms in Lake Erie, kind of the history of harmful algal blooms in the U.S., just to give some perspective on what we're dealing with here in the Great Lakes, and then going into a little bit about just some of the, I guess, high-level understanding of the blooms that we have, not getting too deep into the science, because I don't think that's really necessary at this point, but then looking out to see what does the future look like for Lake Erie, how are we working to monitor it to better understand what's going on in Lake Erie, and then moving on a little bit into hypoxia and hypoxic events or low oxygen events, both in Lake Erie, in the Gulf of Mexico, and throughout the world. So with that, I spend most of my time talking about how algae either harm the environment, harm us, harm our pets, or harm livestock. So I like to start off by having at least one slide about how important algae are for our existence, right? So even though they comprise about 50% of all plant biomass, or sorry, they comprise 1% of all plant biomass on Earth, they produce half the oxygen we breathe, right? So globally, phytoplankton are incredibly important for us being able to survive on this planet as a species. So incredibly important in that regard. They are the base of most aquatic food webs. And I say most because there are some regions, especially in our world's oceans, that are just too deep where phytoplankton can't survive. So obviously if they can't survive, they can't be the base of those food webs. They help fight increasing climate or carbon dioxide concentrations in our atmosphere, especially in our world's oceans, right? And even some of our larger lakes. But when they photosynthesize, hopefully we're all farmers. When they photosynthesize, they take up that carbon dioxide, they convert that into biomass, and especially in the world's oceans and some of our very deep lakes, like Lake Tanganyika over in Africa, when that biomass dies, where does it go? It settles to the bottom, right? And especially in our world's ocean, when it settles to the bottom, it gets sequestered for about 1,000 years. So our oceans are absorbing a lot of the carbon dioxide that we are putting into our atmosphere. But of course, we are here to talk about the negative impacts of harmful algal blooms and the species that cause them, especially in our Great Lakes. So for all the good and all the importance that phytoplankton have, mainly due to human influence, we've created the condition where these blooms, or this proliferation of algae, and the toxins or the compounds they naturally produce that can be toxic to us, increase to concentrations that can become worrisome. And harmful algal blooms, they're not just a freshwater issue, they're not just a Great Lakes issue. They occur throughout the United States. So in our coastal water bodies, right? So coastal water bodies, especially in the red tides, in our estuaries, the brown tides, and then of course in our lakes, rivers, and reservoirs, which are the cyanobacterial or blue-green algal blooms, which is obviously where we're going to spend a lot of our time today. And as I mentioned before, these cyanobacteria, which is the scientific name for blue-green algae, the ones that form in Lake Erie, it's not just a Lake Erie issue. They occur in many of the world's most socially, economically important water bodies, right? So these are very important water bodies on pretty much every continent. So Lake Taihu in China, Matilda Bay, Western Australia, the Han River that runs through Seoul, obviously in the Great Lakes, Lake Okeechobee, if any of you have heard about the state of emergency that have occurred, and in the African Great Lakes. So when you have the American Great Lakes or the North American Great Lakes and the African Great Lakes, that's about 40% of the Earth's available fresh surface water. And each of those systems has many areas that are being impacted by poor water quality, leading to harmful algal blooms. Right? They're also being impacted by invasive species. So our surface water that is so critical to many processes on Earth are being impacted globally by nutrient enrichment, harmful algal blooms, and hypoxia. But before we go too much into the details on Lake Erie, I wanted to take a bit of a stroll back in time because I think it's important to set some historical context. So back in the 60s and early 70s, harmful algal blooms were essentially considered regional events. They were mainly considered marine events. Right? There's a lot of the information from that time, squeaking here, focused on marine events and on primarily marine toxins. So these are the shellfish poisons, so neurotoxic shellfish poison, paralytic shellfish poison, or ciguatera, which is mainly a tropical toxin. Funding was very difficult. We didn't have a national HAB body. There wasn't a national HAB community. So everyone was on their own. But on the other hand, very important discoveries were waiting to be made. And there's one update I want to put onto this map because, again, this focuses on marine, but they should have had a green dot in Lake Erie because in the 50s, 60s, and into the 70s, Lake Erie was also experiencing harmful algal blooms, hypoxia, much like it is today. So looking at the harmful algal blooms from the 50s to the 70s, because of the industrial growth on Lake Erie in cities like Cleveland, Toledo, they were a focal point for industrial growth. But we had relatively few environmental laws at that time. So obviously pollution quickly became out of control, especially from these point sources from industry. And we started to see a lot of harmful algal blooms, even though the science and the data on that is not very well reported or captured. We know that they were occurring, and by the mid-1960s, Lake Erie was considered to be a dead lake due to hypoxia, due to harmful algal blooms. But it also became the focal point for the environmental movement. So many of our environmental laws that we have in place today, like the Clean Water Act and the Great Lakes Water Quality Agreement, stemmed from this early or these earlier instances of HABs and hypoxia in Lake Erie. We were also highlighted in Dr. Seuss's book, The Lorax. If you have a pre-1985 copy sitting on your shelf anywhere, you will see that when he's talking about the hummingfish, the last line in it is, I hear things are just as bad up in Lake Erie, when he's talking about the poor water quality and the hummingfish having to leave. So we made it into his, iconic children's book. But during that time, there were some really important fundamental science experiments that were occurring in the experimental lakes area up in Ontario that helped define a lot of the water management practices we still use today. So this is work done by our Canadian colleague Dave Schindler and his students back in the early 1970s, where this is Lake 226. And these experimental lake areas are extremely important because you are allowed to do whole lake manipulations. So what Dave and his students did is they put a barrier right here, and they seeded this part of the lake with nitrogen and carbon. Then they seeded this part of the lake with phosphorus, nitrogen, and carbon. Obviously, you could tell by the watercolor, bloom occurred, bloom did not occur. That set the early stage for phosphorus being incredibly important for controlling freshwater harmful algal blooms and freshwater phytoplankton community growth. That experiment, those series of experiments, again, helped define a lot of the management strategies we still take today. The science has evolved, and we'll get into how the science has evolved in a few slides. But this experiment, these sets of experiments that were reported back in the journal Science, really set that stage, set the tone. And because of that, we know that phosphorus was a driver of freshwater harmful algal blooms. Lake Erie was considered a dead lake. We had the Great Lakes Water Quality Agreement and the Clean Water Act. The Clean Water Act and the Great Lakes Water Quality Agreement set phosphorus standards, right? So they set phosphorus load standards for Lake Erie, and so that we don't want to exceed 11,000 metric tons per year of total phosphorus heading in or going into Lake Erie, mainly from point source. So the Clean Water Act, is that me? I think it is. Yep, that's me. Sorry. There we go. So the Clean Water Act helped establish regulations for reducing phosphorus from point source pollution. And, oh, geez, that's horrible. Okay, we're going to try to move that up because that's obviously not working. Okay, there we go. All right, so, nope, still there. Anyway, I'm just going to hold it. So, is that me, or what's going on with that? Yeah, if my heart would stop, maybe that would solve all our problems, right? You guys could go out and get some donuts, and then I could just drag me out at some point. Oh, I think you're right. Look at that. We're diagnosing the problem. That's true. Okay, there we go. Sorry about that, everyone. All right, let's try that again. So, because of the Great Lakes Water Quality Agreement, the Clean Water Act, significant improvements to Lake Erie's water quality were being made throughout the late 70s and into the early 80s. But during that time, other regions of the country were starting to experience other types of harmful algal blooms. So, brown tides started to appear in the bays along the east coast of the U.S. And this is incredibly important. So this is Great South Bay. This is a bay that I grew up on. I'm a Long Island native, even though I don't sound like it. The Midwest beat that accent out of me way long ago. But this is the Atlantic Ocean. This is Great South Bay. The reason why this is important is because in the 70s and early 80s, 2 out of every 3 shellfish eaten along the east coast of the United States came out of this bay. Incredibly important shellfish industry. But because of all the overfishing and because of nutrient pollution, you started to see these brown tides occur, which decimated that industry and has never since recovered. We also started seeing red tides from Maine down to Florida, right? Different types of algae comprise those blooms, but we started to see these red tides occur, which were, again, not only did it have impacts to a tourism industry in Florida, which is obviously critically important, but also shellfishing industry up in Maine. So we're seeing social and economic impacts of harmful algal blooms along our coasts, and they're increasing in prevalence, they're increasing in scope and intensity. However, because of the phosphorus reductions, we were able to, this is some work done by Joe DePinto and some of his colleagues back in the mid-'80s, we actually documented that Lake Erie, the water quality improved so much that they considered Lake Erie to be fixed. So at one point in time, we considered lakes to be either broken or fixed. So because of the nutrient reductions in the 80s, we considered them fixed. And because of that, as well as other issues that started to pop up, such as invasive species, so the Dreissenid mussel invasion, our focus kind of shifted away from water quality, and as you can see, we're back into where we were, but for somewhat different reasons. We even convinced Dr. Seuss. So in the mid-'80s, a couple of Ohio Sea Grant researchers wrote to Dr. Seuss and said, hey, Lake Erie's improved, your book's making us look bad, can you do something about it? And he and his editors said, sure. So any print version of the Lorax after 1985 no longer has this line in it. But unfortunately, due to other environmental conditions, sure.
Just curious, back in the 80s, no-till farming came in, ag was given a big star for helping them.
Yep.
I'm assuming there was a lot of urban fixes also.
A lot of urban, a lot of sewer, water treatment plant upgrades, a lot of industrial.
I'm trying to understand how we fixed it back then and now where we're at.
Great question. So that is a great question. I'm going to hold that off till the end, right? Because that does come down to the crux of the management issues that we see today, right? So, but that is an excellent question. We also see that around the country, harmful algal blooms are being reported pretty much in every state. So not only are we seeing different types of harmful algal blooms that we weren't monitoring back in the 70s, but each one of these green dots represents harmful algal blooms that we have seen in Lake Erie and other parts. But really, this entire continent should be green, because it's not just one lake in every state. There's many lakes in every state that are dealing with these cyanobacterial harmful algal blooms. So this is really a nationwide issue. But why have these HABs proliferated? Well, we know that it's, human influence nutrient loading. Urban, agriculture, it just depends on your watershed, where you are, who's influencing that nutrient loading. Loss of key food web predators, as we talked about, mainly along the coasts. Invasive species have altered ecosystems that have allowed some harmful algal bloom species to gain an advantage. Climate change is expanding the range of some harmful algal bloom species and expanding the amount of time that they can survive in different areas. So it's allowing different niches to be filled by different types of algae that weren't able to grow in those regions previously. But also, we're looking harder. Back in the 70s and early 80s, there weren't as many scientists studying harmful algal blooms as there are today. So we can't chalk all of this increase up to degrading water quality conditions, although a lot of it can be. We also have to recognize that we're looking harder. And because we're looking harder, and because we are seeing all these events, we know that the intensity and toxicity of these events are increasing. And this is leading to some recent freshwater events. I don't have to go into the Toledo water crisis. I'm obligated to talk about it because I live in Ohio in every presentation I give. But around the country, we also see that in Lake Okeechobee, there was a state of emergency declared in 2016 and 2018 when the U.S. Army Corps of Engineers, they have to maintain water levels in Lake Okeechobee, so they open up the dams flowing down the Caloosahatchee to the west and the St. Lucie to the east, and they pump potentially cyanobacteria-laden water down these rivers, down these canals, out to the west coast and to the east coast. And in 2016, this occurred right over the 4th of July weekend, where they released a slug of water that was very toxic and very green, and it ended up on the Atlantic Ocean beaches. Right? So again, huge economic impact to that region, especially over the 4th of July weekend. Utah Lake, 130 people that came in contact with that bloom that covered about 90% of the lake in 2016 reported symptoms of what we know to be non-lethal microcystin poisoning, so such as diarrhea, vomiting, headache, rashes, etc. So last year, Salem, Oregon also had a water crisis very similar to Toledo, except this one lasted about 3 weeks on and off, right, where about 150,000 people weren't able to drink their drinking water. So again, this is a nationwide issue where we're seeing social and economic impacts due to harmful algal blooms, especially in our freshwater systems.
Just a question there. Were
Yep.
all those events the same organism, the same species, cyanobacteria?
That is a good question. Utah Lake was microcystis, Lake Erie. For the most part, yes, microcystis was dominant in most of those blooms, although some of them, microcystis tends to dominate in Lake Erie, but some of them had more of a mixed bag, had a mixed population. But that's a good question. And then, of course, the Maumee River algal bloom in 2017. Even though Toledo does not draw its drinking water from the Maumee River bloom, when this occurred in late September in 2017, you couldn't find a bottle of water anywhere in Toledo, right? So there's a visceral reaction to these blooms. There's a knee-jerk reaction from the Toledo water crisis where people in Toledo, still don't trust the drinking water, so there's a lack of confidence in their water treatment system. And also when you see a bloom like this that occurs downtown Toledo, people tend to react, right? So it's not just a environmental impact, but it's an economic impact of people, purchasing bottled water, not being able to drink, or not being willing to drink tap water. So there's additional impacts that occur due to these blooms. But because of all of this, there's growing bipartisan support throughout Congress for drinking water issues throughout our nation. So these are 3 letters. One was to at then President-elect Trump back in January 2017, to OMB Director Mick Mulvaney in November of 2017, and then in December of 2018 to Mick Mulvaney again. Basically, my colleagues and I helped draft this language where we had 47, 53, and 61 lawmakers sign on saying that harmful algal blooms are a nationwide issue and they deserve federal attention and federal funding. So again, this is a bipartisan issue. You see support from both sides of the aisle. But when it comes to the Great Lakes, we see harmful algal blooms in all 5 of our Great Lakes. We tend to focus on Lake Erie, but these blooms occur even in Lake Superior, one of the deepest and coldest lakes on Earth, has seen harmful algal blooms twice in the last 5 years. This is somewhat extraordinary because we often think of lakes like Superior as being somewhat buffered from harmful algal blooms. But when you have the St. Louis River that floods due to these what we call 500-year rain events, it pumps out nutrients into this western basin and around the Apostle Islands. In the last 2 out of the last 5 years, they've seen after these massive rain events, blooms occur along this shoreline. Even though they're not toxic, it's still disruptive to their tourism industry. But again, this is a region-wide issue, but we tend to focus most of our attention on the western basin of Lake Erie for very good reason. And in the western basin, this is work done by Laura Johnson at Heidelberg University and my colleagues at NOAA, Rick Stumpf, and Limnotech up in Ann Arbor. What they do is at the beginning of every season, I'm sure everyone's very familiar with this, they use the cumulative total, what they call bioavailable phosphorus, so the phosphorus that algae is able to use, and they are able to give a prediction of bloom severity, right? So size. We cannot yet predict toxicity. That is a major issue, and that's a major source of research funding for the Great Lakes right now. But what we can do is look at the severity, right? So how large is this bloom going to be? And because of that, because we're seeing this increase in severity, where I should go back and say in 2011 and 2015, we've seen the 2 largest blooms in recorded history occur on Lake Erie. And if you look from 2002 to 2018, if you put a trend line here, you can tell, except for a few years, right, there's always going to be interannual variation due to rainfall, because rainfall drives a lot of this phosphorus loading. We do see some smaller blooms, and even though 2018 was smaller than predicted, we do see a general trend of increasing severity. And because of that, we're always asked, well, can't we just add something to the lake that's going to make the blooms go away? And I put this up there because there are several types of in-lake treatments that are effective at dealing with cyanobacterial blooms in small systems. Right? When you look at a system like Lake Erie where these blooms can be the size of Rhode Island, it's not feasible economically or functionally to try to treat the blooms themselves. The only way we can treat the blooms is to treat what's driving the blooms, and what's driving the blooms is nutrient over-enrichment to western Lake Erie. And it's because these treatments tend to treat the symptom of the problem, not the problem itself. So if you have a small system where you just don't want green water in the summer, you can treat a small system. You can treat a small lake. And for the most part, they can be very effective at removing cells from the water column. But it still doesn't get at why are these blooms occurring in the first place. So these are what I call a Band-Aid to a mortal wound. It doesn't do anything. It just kind of slows the effects of what the actual problem is. So in Lake Erie, we take an integrated approach to studying harmful algal blooms. We use genetic techniques. We use near real-time observations. We use satellite monitoring. We try to understand how invasive species are impacting the blooms, and we try to use the most advanced technologies possible to better understand the ecology, because the better we understand the ecology of these blooms, the better we're able to forecast, predict, and also develop robust management strategies for long-term relief from these events. And I often joke that Lake Erie is probably one of the most well-studied systems on Earth. We have federal, state, academic, and citizen science groups that are out there pretty much from May through October, you can find a boat out there pretty much on any given day taking a sample of Lake Erie water or along the shoreline. So this is a very good collaboration at the federal, state, and academic level working with citizen scientists. So this is a great example for other regions in the nation on how we can work together to understand these blooms, because Bowling Green State can't do it all, Ohio State can't do it all, the federal government can't do it all. We have to work together because it's a big system. But when it comes to blooms, one of the biggest issues we have are that blooms are patchy. So when I look at it from a science perspective, how do I study something that looks like this, right? Because where I choose to sample is going to dictate the results I get. And this is a big issue for studying Lake Erie, is that not only do you have these surface scums that occur, this picture is actually from Lake Winnipeg, but if I were to sample here versus here, if I go out on a boat and take that sample, I get two very different results. And that drives the science and the conclusions that I make. So in order to get around that, we're using these real-time observation technologies, whether it's for water quality in general, for dissolved phosphorus, for dissolved nitrate, or for toxins. We can have these technologies that allow us to take samples without being on the water, and that's incredibly important. And did it freeze on me? Yep, I think it froze on me. Oh, nope, there we go. You scared it. This is incredibly important because if you look at these two graphs, this is phosphorus on the top and this is nitrogen on the bottom. These little black pluses, which are a little hard to see from where you're sitting, but they're right here. This is where we actually go out into a boat on a weekly basis and collect a sample. And all these gray dots and blue dots are where the instruments that we deploy are able to take samples. And that's really important because if we go out on a weekly basis, we can get a broad trend in nutrient concentrations, but we miss peaks that could be very important in driving the ecology and driving the size of the blooms just because we can't afford to get out there every day. So this gives us fine-scale resolution of nutrient concentrations in the lake, which helps us understand the changes in biomass in that system as well. But I do want to point out a cautionary tale that, if the computer lets me, nope. It's a Wednesday morning at 8. I think the computer is a little... There we go. This is important. Oh, whoops. Can you go back one? Okay. Anyway, I'll keep going. The bottom line is that we need to continue to have these on-lake monitoring programs because we often get asked the question, well, why not just use these technologies exclusively? And this is why. Because when you have a deployment, sometimes you just get a bubble. Sometimes you get a bad calibration and you get bad data. So having people out there to go out and ground truth the samples that you're getting from this technology is incredibly important. So we can't rely solely on technology, but it helps us out a lot.
How long is this time series?
This is a year. This is from May through November. So this is just annual. Yep. And we can also do this with pigment, right? So we can track cyanobacterial or algal biomass through these technologies as well. They're a bit more variable just due to the nature of the organisms, right? So microcystis is colonial. It forms, even though it's a single-celled organism, it forms these colonies because it doesn't like, it gets lonely, essentially. So while we can use this to track biomass, we know that there are discrepancies between what we take back into the lab and are able to extract using our lab-certified protocols versus what we see in the field. But they're all very helpful in us getting this high-resolution data. We also know that toxins vary significantly across the basin, and so much so that we know that we can't get out there and sample any one group and be able to capture that. So last year, through some NOAA funding, Ohio State University, Bowling Green State, University of Toledo, and Limnotech all got together. We tried to get our Canadian partners involved, but they couldn't mobilize in time. And we gridded the Western Basin into 4 sections. And in each one of those sections, we took 25 samples each, right, over a course of 2 hours. Right, so all of us were out there from 8 a.m. to 10 a.m., I believe on August 8th last year. And we collected over, I think it was, by the time we were finished, we collected 125 different samples. We went back and we analyzed them at the University of Toledo, or we filtered them, and then we analyzed them for toxins. And what we found was that so this is a heat map of total microcystin, so total toxin, where the green is relatively low toxin, so below detection to about 0.3 micrograms per liter or parts per billion, up to red being the highest. And what you can see here, this underlying map is where the bloom biomass was on that day. All these dots are where we actually took a sample. And it varies. On a given day, the bloom biomass can vary from pretty much non-detect all the way out to 8 micrograms per liter. That is a huge variation in this bloom. And also, toxin concentration doesn't always line up with more biomass, right? So we can't just say, well, there's more biomass, there must be more toxin. This data says that that is not true, right? So not only is there a lot of spatial variability in the toxin throughout the bloom, there is a lot of variability just in the densest parts of the bloom, where you see all this red, doesn't necessarily equal the highest toxin concentration. Actually, the highest toxin concentration we were able to see on that date came out of this region, which did not have the highest biomass. So again, biomass does not always equate to toxicity. But what we do see, and what's very important with this annual monitoring that we've been doing since 2002, is that we're able to see annual trends. And those are really important because seeing those annual trends allows us to create and develop experiments, which then we can test whether or not these correlations have causation, right? So what's driving these correlations? So here on top is nitrogen concentrations, phosphorus, this is phycocyanin or blue-green algae, and this is particulate toxins. And pretty much what we see is that when toxins are elevated during the bloom, it's usually early in the season, and it's usually equated with high nitrogen concentrations. As nitrate, this is nitrate, as nitrate decreases, toxicity decreases throughout the bloom. So we think that there's a link, right? From these data, we think there's a link between nitrogen and bloom toxicity. And this just kind of zooms in on that, where we can see phycocyanin. So these are peaks in bloom biomass, right? So early in the season, early August, late August, and early October. This is microcystin concentration. We have equal peaks of phycocyanin on these two dates, so early August, late August. But when you look at the microcystin concentration, it's around 8. But when you have an equal peak in biomass of cyanobacteria, late August, it's half that concentration. So there's some ecology going on that we need to figure out. Why do we see this discrepancy in toxicity? Because if you're a water manager and you're solely going on bloom biomass, you're going to treat these blooms as the same, even though the toxin concentrations are very, very different. So we conduct experiments. This one's from Sandusky Bay, and I picked it because even though we see very similar responses in the open waters of Lake Erie from Sandusky Bay, this picture, I think, shows it very well, where we have our control bottles. We have a bottle where we take ambient water, so surface water, and we only add phosphorus, and then we have treatments where we add nitrogen and phosphorus. So what you can see here is any bottle where we add nitrogen and phosphorus, not only do we see an increase in biomass, but we see an increase in toxicity. So that links the causation to the correlation that we see. So nitrogen is extremely important in driving bloom toxicity. Phosphorus is very important in driving bloom biomass. Nitrogen is extremely important in driving bloom toxicity. Both nutrients are incredibly important. And this is also important because when we look at the future of blooms in Lake Erie, whoops, got excited there, hit the wrong button. we see that average annual precipitation, this is from the 4th National Climate Assessment, is going to increase several inches by the years 2041 to 2070, right? That's predicted based on various climate change scenarios, but we know that it's going to, especially in the Midwest and the Great Lakes region, it's going to increase. Why is that important? Well, precipitation, the amount of precipitation is expected to yield more nitrogen, right? This is some work done by my colleagues out on the West Coast. This is Anna Michalek's group. She did some work at the University of Michigan before moving out to, where did she go? No, she's in California. But this is work that was, this work was published in Science in 2017, where under 3 different climate change scenarios, the same ones that were used in the National Climate Change Assessment, increased total nitrogen into in the Great Lakes region. So this is increased nitrogen load in the Great Lakes is expected to increase from anywhere between 10. Sorry, what? You lost your mic. I lost the mic. It's still on. Hello, hello, hello. Can you hear me? I can speak up because we're having technical difficulties. So, in the Great Lakes, total nitrogen load to the Great Lakes is expected to increase anywhere between 10% and 20%, which is very important as you come into bloom toxicity and bloom biomass. We know that increase. Yep. Oh, thanks. There we go.
We'll have too many things for you to handle.
I know, that's okay. I got it. Okay. All right, and we're back. In this region, so the near future versus far future, 10 to 20% increase in nitrogen, which means that not only are the blooms predicted to get larger, but they're predicted to become more toxic due to climate change. And this is important because as we come up with nutrient remediation strategies, we have to move away from the phosphorus-only mentality. We have to move into phosphorus and nitrogen control, what we call dual nutrient control, because that is the only way that we're going to be able to drive blooms that are small, right? So all of these nutrient reduction programs aren't to eradicate blooms, they're to reduce the impact, the size, and the toxicity of these blooms. And the only way to do that is to have dual nutrient management approaches for urban and agricultural areas. And finally, how am I doing on time, Greg? Am I good? Perfect. Okay. 20 minutes. All right, great. So I have about 10 minutes, and then we can move into some questions. But the other consequence of eutrophication or increased nutrient loading is hypoxia and dead zones, right? This is obviously a really important issue not only in Lake Erie, but also in the Gulf of Mexico. Right, so hypoxia is when we have dissolved oxygen levels that fall below 4. Anoxia is where dissolved oxygen isn't detectable. So those are just some basic definitions. This diagram here kind of gives you a general idea of you have nutrient-rich waters that flow in, you have algae that grow, they feed, and they die. When that algae dies, it sinks to the bottom where bacteria, they love carbon, they love eating, so they break down all of that biomass that's sinking to the bottom of the system. And while algae produce oxygen, bacteria like to use oxygen. Right, so they end up depleting the oxygen in this region, which can have extremely critical impacts to the benthic life that lives in that region. But hypoxic zones, hypoxic or anoxic areas are a global issue. These are some of the world's most serious hypoxic zones, and as you can see, a lot of them are along the coastlines where harmful algal blooms occur, where these areas are being most impacted. So this is a global issue. This is the Gulf of Mexico hypoxic zone, one of the largest or the largest in the world. And that is obviously driven by nutrients coming from the Mississippi River out into the Gulf of Mexico. And this zone obviously can increase or decrease depending on what the river is doing at any given time. So this is incredibly important because, again, when you look at the Gulf of Mexico and when you look at their industry and when you look at their income, a lot of it is driven by commercial fishing. So when you have these dead zones, it impacts that industry significantly. So the larger the bloom, or the larger the zone, obviously the more economic impact it's going to have to that region. And in some systems, these hypoxic events can cause fish kills, right? So when you have a system that has a chronic algal bloom, one of the things that algae do, especially the reasons why we see the scums and we have those impressive satellite images, is because these blooms like to float to the surface. When they float to the surface, they block light. When they block light, they kill plants that live on the bottom. That's extremely bad for systems for a couple of reasons. One, it takes away critical habitat for juvenile fish to live and survive. Two, it creates a condition where the sole oxygen producer, right, the sole biological oxygen producer is the bloom. So if the bloom were to die, and to rapidly die just due to change in temperature, various other reasons, or if you add an algicide, which kills the bloom, all of that carbon then gets consumed, all that bacterial respiration consumes all the oxygen, and you can have fish kills. And these are some samples I took when I was a grad student on Long Island, and you know oxygen is low when American eels are dying, right? Those things can live in anything, right? They live in sediment. They live in very low oxygen environments, so when they're dying, you know oxygen levels are very low. We don't tend to see this occur. These types of fish kills occur in Lake Erie. Why? Because Lake Erie's big and fish can move, right? A lot of these events are, when it comes to harmful algal blooms, are constricted to these small systems that are closed where fish can't go anywhere. But in Lake Erie, we have a dead zone that forms every year, and it's being impacted by algal blooms, but not necessarily the algal blooms that you're thinking of. So the Lake Erie dead zone formation tends to kind of start in June and really peak in September, mainly in the central basin. And one of the reasons why is the bathymetry of lakes. So how the lake is shaped also impacts where and how these dead zones form, right? So annually we have a dead zone that occurs in the central basin. Right, so this is the western basin where the blooms, the summer algal blooms, these cyanobacterial blooms occur. In the central basin, we see this hypoxic water, and that's also an issue for water intakes because they have to deal with treating this hypoxic water if you have a situation where this low oxygen water were to get pushed into the area, especially around Cleveland, of their intake. Right, so this is, even though it's not the water issues they deal with it's not harmful, it's the perception of bad water can be equally detrimental on a community as actual poor water quality. And what we know is that it's not the summer blooms that's driving the hypoxia in the central basin. It's actually winter algal blooms in Lake Erie that drive this, that drive the dead zone. So this is a picture from the Canadian Coast Guard icebreaker Griffin. We were out there doing some winter work a few years ago, and as you can see, well, maybe you can't see, but in this picture, Lake Erie is brown. And at first we thought that that was just mud, but it's not. It's diatoms. So these winter and spring diatoms are a brown algae. They have fucoxanthin, which makes them look brown, which then makes the water look brown. These winter diatom blooms are incredibly important for the health of Lake Erie because they help, these winter and spring diatom blooms, they help feed their healthy algae. This is mainly Algae. They help feed the diatom, or they help feed the zooplankton. The zooplankton help maintain a healthy fishery in Lake Erie. So these blooms are incredibly important, but they're also, all of this biomass is then being transported to Central Basin, where eventually it's dying and settling down to the bottom, which is causing these dead zones. And eutrophication can be making these winter diatom blooms more intense than they might have been previously. So again, Lake Erie doesn't go to sleep in the winter. There's still a bloom. If you break through the ice in the winter, you will find very dense blooms of Algae underneath the ice. It's incredibly, it's a really cool organism, but it's driving this summer hypoxia, right? So it's not the summer algal blooms, it's actually the winter algal blooms that's driving hypoxia. So with that, I did want to leave some time for some questions. So I can take those now. I can start with the one that this gentleman asked earlier, and we can go from there, unless anyone else has a burning question that they have to have answered first. I'm happy to do that too.
Just a question, Tim, that ties into the question
Sure,
here,
sure.
and that is the 1980s and the species at that point of
Sure.
cyanobacteria versus now.
Right.
Are they
So that's a great question. So the blooms that occurred back in the 60s and 70s were more of a mixed community. They weren't as dominated by microcystis as the blooms are today. So the main toxin producer in the open waters of Lake Erie is microcystis. In the tributaries like Sandusky Bay and Maumee River, it's Planktothrix. They still produce the same toxin, but they're different organisms that have different niches. Back in the 60s and 70s, because it was mainly phosphorus, we had nitrogen-fixing cyanobacteria like Anabaena, Aphanizomenon dominating. So they were, even though microcystis was there, we were able to detect microcystis and the genes, so the genetic infrastructure that cells need to produce the toxin. We're able to find those back into those earlier blooms, but microcystis was not dominant. So that's a great question.
Do you want to ask your question again, Albert? Who had that
Well,
question?
yeah, just a question. Okay, so the lake was cleaned up, if you will, at better water.
Yep.
In the mid-80s.
Yep.
What?
So we had a couple things. One, we had the main nutrient load shift from point source to non-point source. So point source obviously being industry, wastewater treatment, places where we can see. So that shifted. What likely drove microcystis to be dominant, so microcystis can't fix its own nitrogen. It can't make its own nitrogen. It needs nitrogen. So as back in the 70s, it was mainly a phosphorus-driven, right, a pollution issue. We see that from the 80s and the early 90s, we see nitrogen or nitrate concentrations increase. So it's increased nitrogen concentrations in the rivers. Those are starting to taper off a bit. Increased phosphorus, again, even though total phosphorus hasn't increased, the proportion of the total phosphorus that's made up of dissolved reactive or this bioavailable phosphorus has increased. So both of those, so nitrogen, increased phosphorus, but also we had the zebra mussels and quagga mussels. And we don't fully understand the impact that those had on the ecology of Lake Erie, because when they first invaded, so in '85, they were first seen in Lake Sinclair. I think it was a few years after that they were in Lake Erie. We don't, because a lot of the monitoring programs were somewhat disassembled because we said, oh, Lake Erie is cleaned. We started to look at other issues. We don't fully understand how the invasion of the dracenids, the zebra mussels primarily, impacted the water quality. But we do think there was some, but mainly it has to do with the shift in nutrient source, increase in nitrogen concentrations coming into the lake, and possibly the invasive species.
Well, just to add to that back in the 80s, it was particulate phosphorus that was the huge issue,
Yes,
which
yes.
is where no-till came from, to try to keep the soil and dredging and keeping the soil on the land. And since then, there's been the increase in dissolved reactive phosphorus because we're getting a higher conversion in the soil from particulate phosphorus to dissolved reactive phosphorus. And no one seems to be able to answer that question as to why is the soil now.
So I think there's some work going on at Ohio Northern that's potentially linking that.
Yeah, there's lots of theories. but no one-
Jay, you look like there's a potential link to increased glyphosate usage on soils that, and I'm not a soil. I am going to say I'm speaking somewhat outside my expertise based on what I've read. If I know that there's work going on this topic here at this university, so if there's anyone in the crowd that's working on that, feel free to correct me at any point. But from what I've read, glyphosate, the increase in the use of glyphosate on soils has allowed for more of this dissolved phosphorus to wash into the wash into the rivers, right? The chemistry of that is something where, again, I'm not a soil chemist, so I'm not going to try to speak on that, but I do believe that there is a theory that links the increased use of glyphosate to this increase in dissolved reactive phosphorus coming off the soils into the river. So it's, instead of it being total, bound phosphorus coming in, we've been able to keep that total phosphorus level below that 11,000 metric ton per year threshold, but it's that dissolved piece that's increasing, and that's the current theory from my understanding. But I'm happy to be corrected on that. I'm always happy to be corrected.
That's one theory.
Yeah.
About 10.
Yeah. Well, that's the most recent one that I've been reading about. So, yes.
It seems to me as if there are parallels here between this issue of toxic, harmful algal blooms and perhaps the whole issue focused around climate change. We know climate change is occurring. We know harmful algal blooms are occurring. Controversy surrounds human-caused climate change, human-caused algal blooms. Could you take us back a little bit further than the 60s into the more of a longer historical, perhaps geologic look at harmful algal blooms in Lake Erie, and I'm thinking specifically of stromatolites. I'm thinking specifically of Oliver Perry's journals, which state extensive dysentery amongst his men as they're fighting the Battle of 1812. Take us back longer and tell us about how these organisms have been in this environment.
Sure. Well, we know that cyanobacteria are a natural part of the community. We know that the western basin of Lake Erie is shallow and warm and is one of the reasons why it is such a kind of a breeding ground for these types of events. What we don't have is very good scientific records of things, of blooms like that. We have these kind of hearsay events, right? And we've seen that all the way back through time, even if you want to go back to Genghis Khan, right, has, saying his men drank green water and ended up falling ill, right? So we have all of this anecdotal evidence of blooms occurring. So that's, again, we know that harmful algal blooms do occur naturally. So these are natural phenomenons. We do know that humans are making, due to our activities, we are making these blooms worse. We're making the frequency increase. We're making them larger. Again, some algal blooms are extremely important and very healthy, right? So the blooms that occur in the North Atlantic are extremely healthy. They need to occur. The blooms that occur in the wintertime need to occur to maintain a healthy system. It's the other blooms, the other guys, that are the issue. So when it comes to the ecological history of Lake Erie and blooms within that, we don't have a very good understanding. We do know that there were likely blooms that occurred naturally, but again, the western basin of Lake Erie, shallow, warm, but also at that time we had the Great Black Swamp, which was acting as a gigantic filter for Lake Erie, sequestering a lot of the nutrients in that area. So it's land use change as well as development, everything else that we're doing. And not that those are bad things. That was natural progression, right? But it has led to the current state of events.
Would you happen to say the pores in water in an uncivilized area?
So that's a great question. There was a paper that came out about a year ago that said that even back in Roman times they were finding, just due to Roman civilization and settling of areas, they were finding, decreased water quality. Could you find these blooms in a forested area that had no human impact? Sure. It's absolutely possible, because there are some lakes that just have naturally high internal phosphorus loading. And if you have that, you can definitely see a bloom, right? So based on the bathymetry, so the shape of the lake, what's coming in around it, what's around the shore, why not? I mean, it's absolutely possible. And we do sometimes see that where there are blooms in areas that aren't necessarily, human influenced, right? So that's where we get into that these are natural. Some of these events can be naturally occurring just due to the state of some lakes, right? And we're not trying to control all of nature. We're trying to limit our impact on influencing these events in nature, right? So it's definitely possible.
And there's a question here. Would you have an opinion on sources of phosphorus, nitrogen as to comparing point source pollution and non-point source?
So comparing point and non-point of nitrogen and phosphorus. So I'm at a primary agricultural meeting, so this is going to be a good conversation. That's right. That's right. It's yes. I study the downstream effects, but what we do, all signs indicate that it's mainly right now a non-point source issue. Both nitrogen and phosphorus have shifted from point source to non-point source. Not that it's a solely non-point source issue, but especially in the Western Basin and in the Maumee River watershed, it's primarily agricultural, and that's just, it's not demonizing anyone. I like to eat food, so I like that you grow food. I really like to eat food, but that we're not saying that urban does not contribute, but the percentage that urban contributes, if we were to focus solely on urban, would not have an impact on what we see out in the lake.
Do you have an opinion on percentages?
For nitrogen, no, I don't. I can't remember them off the top of my head, and I don't want to say something that is incorrect.
I'm talking about for non-point versus point source.
Well, yeah, it's 85. Yeah, that's the number I had rolling around in my head, but yeah, I haven't gotten asked that question in a while, so I wanted to make sure Yes.
We had a communication with the seed industry that we need to fundamentally change the corn plant because nitrogen is linked to the toxin load, and we're headed to greater rainfall, greater rain events, eutrophication. Do we fundamentally need to change the corn plant so that we fix nitrogen itself, or that we can have greater nitrogen use efficiency to help prevent that?
No, I personally have not, because again, I'm aquatic, so once you get on, I think that's an interesting idea, but I haven't had any direct contact with seed manufacturers on that topic, but that is interesting. That's why I like coming to these conferences, because I learn a lot. Yes.
Well, and again, this is just, I'm not from here, but I know what's happened, across the Corn Belt. We've also brought some new land into production. Small grains have decreased, so we have more corn and soybeans. And just maybe not a whole lot percentage-wise, but was that enough to, cross the tipping?
Again, that's an excellent question for an agricultural scientist in the crowd. That's not my area of expertise, so I don't want to. I apologize, but I can't really speak to that intelligently, so I don't want to speak to it all.
All right. I think that's going to have to be the last question.
Thank you very much.
Thank you very much.